the biodiversity, and then we studied different types of biodiversity, and then how to measure the biodiversity and scales of biodiversity. So today our topic is genetic diversity. Genetic diversity is the variation in genetic composition among individuals of a population or a species. Okay, so variation in genetic composition of among the individuals of a species or within a population is basically the genetic diversity. Genetic diversity is also said as total number of genetic characteristics in genetic makeup of a species. So in genetic makeup, the total genetic information or total genetic characteristics are the basically it is the genetic diversity. So any variation among the genetic composition or among the genes of any in a community or within a population or within a species is genetic diversity. And a large genetic pool indicates that there will be higher genetic diversity, and a large genetic pools means that there are high number of there are more number of individuals, and there is more variation among the uh, among that genetic pool. And with this variation, there are increased chances of biological fitness and survival. So their survival chances and their fitness chances among the community is high. Among that particular ecosystem is their chances of survival are more as compared to a genetic pool which is smaller. And genetic gene pool or genetic pool, we already studied this in our previous classes in detail. It is basically the collection of all genes and all type of alternative forms of genes in any population. So gene pool or genetic composition is both are basically same. Collection of all genes, collection of all genes, whether there are variation or not. So the collection of all genes and all the collection of all genes and all the alleles in a population is called as gene pool. For example, you have a population of cows of different colors. The colors are caused by different variation of same gene. All the different variation of the same gene or gene makeup is the gene pool. And it doesn't bother with the frequencies, but a variant is either present or not, whether a variant is either present or not. The composition of population's gene pool can change over time through evolution. We studied this in detail that the composition of gene pool can change over time. The result is a gene pool that is altered to be attuned to the needs of population specific environment. And we studied the example, the migration of human population from equatorial regions towards northern climates, where they were exposed to relatively low amounts of sunlight, and this resulted in change in skin color over time. And change in skin color is due to vitamin D absorption. Okay, then we studied the causes of genetic variation. There are different causes of genetic variation, but the main cause is genetic variation, or generally referred to variation in genes between different individuals of a population. Genetic variation is important for survival and adaptation of a species, as it helps in terms of natural selection and evolution. We studied the natural selection. Natural selection is basically the survival of the fittest. Those genes which are more in number, or which are more susceptible to environment, more resistant to environment, those will be transferred to the next generation. So genetic variation are caused by the mutation, genetic drift, gene flow, and non-random mating and natural selection. Okay. Okay. So basically, mutation of all these, mutation is most important because genetic variation. These are most of the genetic variation. These are caused by mutation. In mutation, we have two types of mutation: gene mutation and chromosomal mutation. A small change affecting the nucleotide sequence of one gene. So gene mutation is basically a small change that affects the nucleotide sequence of one gene. The two main types of gene mutations are point mutation and frame shift mutation. In preceding slides, we will discuss about the point mutation and frame shift mutation. And the other cause is, the other type of mutation is chromosomal mutation, substantial alteration of genetic material of an organism. It is basically a change in structure of the chromosome or either the number of chromosome. And in chromosomal mutation, we have deletion, depletion, insertion, inversion, and translocation. Insertion is the addition of a single or very few nucleotides in DNA. Deletion is the removal of single or very few nucleotides in DNA. I'll not go into the detail. You already know these things. We discussed this in our physical class. So substitution is the replacement of single or very few nucleotide in DNA sequence with other nucleotides. These changes lead to point mutation. Point mutation can be a silent mutation or a missense mutation or nonsense mutation. While insertion and deletion, they can cause frame shift mutation. In silent mutation, we do not have an effect on cell metabolism because the change in codon doesn't lead to the change in amino acid. Okay, so it is a small change which doesn't affect the cell metabolism or the protein synthesis. So it is called as silent mutation. And there is change in missense mutation. Code for different missense mutation code for different amino acids. It result in altered protein, but the protein is still functional. So that is missense mutation. And nonsense mutation code for it codes for the stop codons, which leads to the producing of non-functional shorter protein. Then we have frame shift mutation. As we said, it is caused by deletion. There are different types. In frame shift mutation, insertion or deletion, it occurs with insertion or deletion in the genome that are not in multiple of 3 nucleotide. As a result of these insertion or deletion, the reading frame of mRNA is changed, which result in protein with different amino acid sequence. So if frame shift mutation or if mutation or change occurs at the start of the gene, it will produce totally new protein. Or if it occurs at the end of sequence, there will be change of only few amino acid. Over here we have example that if it occurs, then totally new protein is produced. And if it occurs at the end, then there will be change in the amino acid sequence. Chromosomal mutation. In chromosomal mutation, we have deletion, inversion, insertion, and translocation. Deletion is the removal of large chromosomal region, and which leads to the loss of genes within that region. Inversion is the reversing orientation of the reverse orientation of the chromosomal segments. Deletion is the removal of a chromosomal region. Inversion is the reversing of the orientation of chromosomal segments. Insertion is the addition of material. Addition of a material into the chromosomal region through a non-homologous chromosome. And translocation is the interchange of genetic material between the non-homologous chromosome is the translocation interchange. Okay. अच्छा. Duplication. It leads to the multiple copies of chromosomal region, increasing the number of genes located within a region. And these can be due to unequal crossing, mistake in DNA replication or DNA repair mechanism, retrotransposition, and or the whole genome duplication. Then we studied another cause of the genetic diversity. First one is mutation. The next one is genetic drift. Genetic drift is basically change in the allele frequencies within a small population from generation to generation that occur due to chance event. Okay. Uh, and that chance event that can be natural catastrophe or any natural मुझसे लिखने नहीं हो रहा लेकिन we studied this thing in detail. So genetic drift is a chance event with which a change occur in the allele frequencies within a small population. In each generation, some individuals may just by chance leave behind a few more offspring than the other individuals. The genes of the next generation will be the genes of lucky individuals, not necessarily the healthier or better individuals. So it doesn't depend that whether dominant allele or the best fitted alleles will be transferred or the weaker alleles will be transferred. So it is a chance event. Only lucky alleles or lucky genes that will be transferred. The lucky one which survived the chance event or natural catastrophe that those genes will survive and those will be shifted to the next generation. And genetic drift, it reduces the genetic diversity. So as it is a chance event, we don't know that whether which gene will be shifted, whether the dominant allele will be shifted or the recessive allele will be shifted. So if all of them are dominant allele, so there will be less genetic diversity. If all of them those survived, they are recessive, so there will be less. It will reduce the genetic diversity. Then we discuss the types of genetic drift that we have. Two types: bottleneck and founder effect. Bottleneck effect of genetic drift and founder effect of genetic drift. So any event that occur, the bottleneck event, so those genes which transfer through the bottleneck, they will survive. So we can compare the pre-bottleneck effect and post-bottleneck effect, the gene variation in pre and post effect of that catastrophe. The founder effect: if a population migrate or if a population is isolated, if a population migrate to a new area and they start their new generation, so in the later generation there will be only those genes will be transferred which they have, which the founder population they carry. Only those genes will be transferred that the founder population carries. So what is bottleneck? Over here I have explained. A population of bottleneck or genetic bottleneck is a sharp reduction in size of a population due to environmental events, famines, earthquake, floods, fire, disease, droughts, or other human activities. Such event can reduce the variation of gene pool of a population. Thereafter, a small population with smaller genetic diversity remain to pass our genes to the future generation of offspring through sexual reproduction. genetic diversity remain lower increasing only when genes flow from another population occur are very slow increasing with time as random mutation occur. So it is basically only those population which can survive the event or which can survive the environmental events, those genes will be shifted to the new population. Over here, I gave the example of Californian seal. In initial, its population was 100K, but due to human activities, with the developmental activities and human activities on the seashore, their population reduced to only 50. In 1800, it was 100K. In 18, within this span of I think 60 to 70 years, it reduced, the population just reduced to 50. When humans saw that the population is reduced, the Californian seal population is reducing, so they started conserving those seals, and nowadays the population is almost 150K. So the human activities due to developmental events, due to environmental, developmental events or developmental works, the population of the Californian seal they reduced, and then after conservation, the population is now increased, but very less genetic diversity. Only the founder's population of 50 seals, they transferred the genes in those 50 seals, those genes are transferred to the next generation. The founder effect is the loss of genetic variation that occur when a new population is established by a very small number of individuals from a larger population. And we have the example of Huntington's disease, which occur in South Africa. After much research, scientists were not only able to discover the mutation that causes the devastating disease, but also trace back it to the founder herself. 200 years ago, a woman with 10 children, of all whom in the area to raise their own children, unfortunately, Huntington's is a dominant allele, meaning that if one parent carries it, there is 50-50 chances that it would be passed to the offspring. This disease has been able to persist for long because natural selection no longer acts after reproduction, which is when Huntington begins to take its effect. So the parents, the founders, they have the alleles of Huntington disease, and it transferred to the new generation. And as it was a dominant allele, so it was there was chance 50-50% chances that the offspring they will have this allele. Then we have natural selection, the process whereby organisms better adapted to the environment tend to survive and produce more offspring. Okay. Organisms better adapted to their environment tend to survive and produce more offspring as a result of natural selection. The organism in a species with more adaptable characteristics increase with each generation. So in every generation, those alleles or those genes will survive that are more adaptable to the environmental condition. Therefore, natural selection modifies the originally random variation of genetic traits in a species so that the alleles that are beneficial for survival increases, while alleles that are not beneficial, they will decrease. So natural selection over here in this diagram, you can see the natural selection. So those alleles which are more beneficial for survival, their number is increased, while those alleles which are not beneficial, their number is decreased. Then we studied the difference between genetic drift and natural selection. Natural selection is non-random process. So any allele that is beneficial, that will only those alleles that are beneficial for survival, those will be retained, and the other alleles which are not beneficial, those will not be retained. While genetic drift, it is a random process. Any allele can be um, can survive after the environmental event or after the any natural catastrophe. While natural selection, it occurs due to environmental challenges. Within an environmental event, only those alleles will be survived which can uh, resist the environmental challenge. While in genetic drift, it is a chance event. It doesn't show that whether beneficial allele will be survived or non-beneficial allele will survive. So it is a chance event. Any allele can survive during the genetic drift. Natural selection, it is directional. Only the alleles which are beneficial or which can resist the environmental challenges, they will survive. And it is genetic drift, it is non-directional. The beneficial one can, or the non-beneficial one. Anyone, the positive or negative allele can survive. So it is non-directional. It can go in any way. The diseased allele, whether the diseased allele or healthy alleles, they will survive during the genetic drift. So any allele can survive during genetic drift, but during natural selection, only those alleles which can resist to environmental challenges, which are beneficial, those will survive during natural selection. And only the adaptive traits will be survive. And in genetic drift, non-adaptive traits, they can also survive. Natural selection, it ends up with the survival of the fittest, while during genetic drift, important alleles may disappear during the genetic drift event.